Very, very dramatic indeed. Good morning. It's great to see everybody. It's great to see friends. It's been a year since I gave a talk, so I'm trying to get my way around the mic over here. Thank you to the organizing committee for the opportunity of presenting Iveric Bio to you. Our disclosures are in our filings. So with age-related macular degeneration, what I would say is probably the most important thing that we've learned physiologically in regards to macular degeneration, that it is very simple, is that it's a chronic healing process. It's a chronic inflammatory process where complement is activated. And that's probably the most important finding. And that's true from the time that drusen appear all the way to geographic atrophy. What we've also learned is that the progression of geographic atrophy is remarkably stereotypic, regardless of your demographics, regardless of your risk factors. It always progresses the same way, which is that it starts outside the fovea and then progresses relatively rapidly. And I use that word rapidly deliberately because it's not a matter of years or decades, it's a matter of a few months in a circumferential fashion around the fovea, and then hits the fovea and then tends to slow down. So remarkably stereotypic. We also know that it's the leading cause of blindness in this country. The estimation here is that it's about approximately 1.5 million people, but undoubtedly that's an underestimation. We also know that it's increasing throughout all demographics around the world. So this is an increasing market, a rapidly growing market as we live longer. So where does Zimura fit in? And why is Zimura important? This is the complement pathway. It looks complicated, but when you really look at it, it's not so much complicated as it is redundant. And redundancy in nature, as in life, is really a reflection of importance, and it is very important. It really keeps us alive and keeps our endogenous and exogenous pathogens away. So when you look at this, what you see is three entirely independent pathways that converge on C3, and then on C5, and then split out into two different pathways that kill cells, the membrane attack complex and the inflammasome. So if you look at this intuitively, you'll see that the higher up you inhibit in this system, the more likely your inhibition is to be overcome by the redundancy. After all, that's why it was designed that way. So when you look at previous drugs that have failed, in a gene-agnostic patient population, it's important to emphasize that in a gene-agnostic patient population, you'll also see that these drugs tend to be fairly high up, and possibly the inhibition was overcome by the redundancy. So that leaves behind C3 and C5. Now, we firmly believe that C3 inhibition works, as has been shown recently. However, we also believe that it's important to keep that C3 loop intact if possible. It happens that it's very, very important for photoreceptor cell metabolism, opsonization, neuroprotection, microischemia, perfusion, et cetera. So while C3 inhibition, we believe, does work, we also believe that C5 inhibition with Zimura is both safer and stronger, as I will show you here. So we have a phase 3 program that we call GATHER. There are two pivotal trials. One has already completed and published, which is GATHER1. GATHER2 has finished recruiting. So let me show you some data. So the GATHER1 study is an 18-month study with a primary endpoint at month 12. And again, this has been completed successfully. It's the first successful trial in geographic atrophy as a phase 3. To cut to the chase, you'll see here that in both doses, the 4 milligram dose as well as the 2 milligram dose, it was statistically significant, quite impressively, and decreased the rate of growth of geographic atrophy by approximately 30%. This is an efficacy profile of the 2 milligram dose that we plan to develop. On the top, you'll see the prospective calculations, and below the retrospective calculations. And what you see in the top two, let's start with that, is really a remarkable consistency. What you see is that there's a delta with the very first measurement. And with each measurement, that delta gets bigger and bigger and bigger, with the biggest delta being at the end of the study. This is true in the 2 milligram dose, as I've shown you over here, whether it's calculated with square root transformation or non-square root transformation. It's also true, although not shown here, in the 4 milligram dose. In the below two panels, what you'll see is a retrospective calculation, a post hoc analysis of earlier stages of geographic atrophy. On the left, the progression of iRORA, or incomplete RP and outer retinal atrophy, to CRORA, or complete RP and outer retinal atrophy. And you can see that that is also slowed down. And on the lower right, you'll see an even earlier stage, which is from drusen to iRORA or CRORA. Again, as far as I know, for the first time, that's also been shown to be slowed down. So when you look at this, what you see is a remarkable consistency in the efficacy profile. It is always the same, which is that there's the initial separation with the delta getting bigger and bigger and bigger over time, with the biggest delta being at the end of the trial. And that's true regardless of the dose, regardless of how it's calculated, and really regardless of the target. So this consistency should give us a great deal of confidence as to the robustness of the data. As far as safety is concerned, there were no Zimura reported adverse events. There was no inflammation, no endophthalmitis. And when you look at the conversion to choroidal neovascular membrane formation defined traditionally, what you'll see here is that the conversion with the 2 milligram dose was, we believe, a very acceptable 9% at 12 months and 11.9% at 18 months, so 2 more patients. This is our GATHER2 trial. This is the last part of the phase 3 program. We have recruited 448 patients already. They've been randomized to receive the drug on a monthly basis versus sham. A pre-specified endpoint is at month 12, and upon achieving that, we intend to file for approval. What we've announced recently is that we have a target in GATHER2 of making sure that the injection fidelity is well above 90%. So what is injection fidelity? Well, that is, in our opinion, the ultimate measure of patient retention. It is actually the total number of injections administered divided by the total expected. It's about transparent as you can possibly get as far as retention is concerned. And we've publicly announced that our goal here is to make sure that this is above 90%. And why is that 90% important? It's important because in GATHER1, the injection fidelity was actually 87%. So if we're able to deliver the robustness of the data that we did in GATHER1 with an 87% retention rate or injection fidelity, we're more than confident that we'll be able to do as well, if not better, in GATHER2. So in summary, if positive, we expect GATHER2 will be the final pivotal trial required for FDA and EMA approval for geographic atrophy. And as you may know, we have a SPA agreement with the US FDA, the first of its kind for this disease. Zimura has the potential to impact earlier stages of AMD, and we believe we're well positioned to expand Zimura's indications, build an AMD franchise, and subject to regulatory approval, commercialize Zimura for geographic atrophy. I want to thank you for your time and for your attention.